Yes, hello guys, welcome back to the biotechnology lessons. So today we would be actually discussing the next part in biotechnology, which is genome extraction process. Just to help you actually make a connectivity with why we are doing it, I would basically give you a quick recall of what we did in the previous lecture. Number one, what exactly is biotechnology? That is use of some living organisms in their natural form or in genetically modified form to actually make them beneficial for humans, right? So if you are using a natural organism for human benefit, that is called traditional biotechnology. But if you are modifying an organism to actually make it useful for human, that is called modern biotechnology, in which modification in the genome of the organism is called genetic engineering. Now, the purpose of biotechnology was basically to produce what? A product, a protein product of human interest. So let's say that protein product can be anything. It can be a hormone, it can be an enzyme, it can be just basically a structural protein or anything of that sort. So if you want to produce a protein of human interest, you would basically require what? A gene that can actually code for that product. Fine. And where would you get the gene from? From some organism, obviously, who is naturally carrying that gene. Okay, so today's lesson would be actually focusing on trying to extract the genome of an organism who is the natural source of our protein product, of our desired protein product. Okay, so just a quick recall. Biotechnology, the purpose over here is to basically obtain a protein product of human interest. And most importantly, obtaining the protein product of human interest in large quantity. That is what we want to do over here. A protein product of human interest, which can be, as I told you, a hormone, an enzyme, or any other protein of human interest in large quantity. Okay, so if you want to get a protein product of human interest in large quantity, you would be requiring a gene that can actually code for this protein product. And where would you get the gene from? From some organism, an organism who naturally carries that gene and naturally also produces the protein product which is of our interest. But then I told you in the previous lecture that why don't we just get the protein product from that organism only if it is producing it? Because that organism is producing the protein product of human interest, but not in large quantity, only in very small quantity, maybe for its own uses only. But we want that product in high quantity, large quantity. That's why we would basically need that gene from that organism which can code for our desired protein product. Okay, so the purpose is to obtain a protein product of human interest in large quantity. For this, we need a gene which can be called the desired gene, with which we will be actually coding the gene that will be coding for our desired protein product. And where do we get the gene from? This is obtained from some organism who naturally produces this protein product. This gene of human interest, which will be coding for the protein product of human interest, this is obtained from some organism who naturally produces this protein product. But in how much quantity? Low quantity. While my aim is to get this same protein product in high quantity, so I would be actually taking out the gene and then using this gene to produce the product in high quantity. So that, I think, establishes a link of what we are going to do today here in this lecture. What I need over here is a gene from some organism who is the natural source of my protein product. But then it's obvious that you cannot get a single gene from an organism. You have to get the entire chromosomal content or the genome content. Once you get that chromosomal content or the genome content, you can break it down into smaller pieces and then identify one small piece or one small gene that would be your gene of interest, and then use it further in biotechnology. Okay, so with this, let's go forward. What our aim today is to obtain a gene, but then as I told you, obtaining a gene alone is not possible. That is why we have to get a entire genome first. So genome extraction process. So let me take you forward now. Genome isolation or extraction, we can say, is to be done from an organism who is the natural source of our desired protein product. So this is the organism whose genome we want to obtain. So genome isolation or genome extraction, we can say, is to be done from an organism who's a natural source of our desired protein product. Why do we want the genome? So that we can break down this genome into smaller fragments. And what are the fragments of the genome? Genes. And then identify that particular gene which will code for our desired protein product. So by the way, that would be actually the next step, breaking down the genome into smaller pieces or the gene fragments. So right now, today we'll be actually focusing on genome isolation or genome extraction from an organism who is the natural source of our desired protein product. So let's see what exactly is the procedure of genome isolation or the mechanism of genome isolation. Now, mechanism of genome isolation is a set protocol which is very easy to perform in laboratory. The first thing that you need to do while trying to obtain the genome of an organism is take a few cells of the organism whose genome is to be isolated. So for example, you are actually trying to produce a protein product that is produced by some bacteria naturally, but in very less quantity, and you want that product in mass quantity. So what you need to do is take out the genome of the bacteria and then break it down and identify the gene that would actually help you obtain the same protein product in large quantity. If you want to obtain the genome from, if you want a protein product that is produced by maybe an yeast, so you'll require the yeast genome now. If you want to obtain a protein product which is naturally produced by a plant, but you want that product in high quantity, you have to take out the genome of that plant, obviously. Okay, so what we need to do is take a few cells of the organism. Like if I want to isolate a genome from a plant, just take a bunch of plant leaves. That is basically what? A group of few cells or tissues of the plant. Okay, so what we do is take a few cells of the organism whose genome is to be extracted or obtained, and we basically grind these cells. Like, put the bunch of plant leaves in a grinder and just simply grind it. You'll get a paste. From that paste, you can obtain the plant cell genome. Okay, so take a few cells of the organism whose genome is to be extracted and grind these cells and dissolve them in buffer solution. We are not going to use simple plain water over here. All the solutions which will be actually used to dissolve whatever material we are using in the genome isolation, that will be always buffers, because we want the pH to remain constant during the entire genome isolation process. The reason for that is there will be actually a lot of steps in this mechanism of genome isolation which are enzyme dependent, and since enzymes act only at a fixed pH, we'll be using buffer solutions instead of pure water. So what we have done over here is take a few cells of the organism whose genome is to be extracted, maybe grind them and make a paste out of them, and put that paste in a buffer solution. Here's your buffer solution, and this basically is, let's say, the grinded cells whose genome is to be isolated. Okay, so if I want to mark it over here, these are the grinded cells whose genome is to be isolated, dissolved in what? Buffer solution. The first step now after dissolving the cells in the buffer is taking out all the components of the cells in the buffer by breaking down these cells. So how do you actually break down these cells which are dissolved in the buffer over here? This depends on what type of cell these are. If these are animal cells, what you need to do is just high-speed rotation. High-speed rotation of this test tube is going to basically rupture what? The cell membranes of the animal cells. And once the cell membranes are ruptured, all the contents of the cell come out. So with animal cells, it's not a big problem, because animal cells just have simple cell membranes, and cell membranes can be easily ruptured by high-speed rotation. The problem is if you are trying to extract a genome from a bacteria cell or from a plant cell or from maybe a fungus cell, because they have got what? Cell wall around the cell membrane. So we need to rupture the cell wall and then the cell membrane, and then only the cellular components will come out where in the buffer. So if these cells whose genome is being extracted are animal cells, then as I told you, it's not much big of a deal. High speed rotation which is also called what? Centrifugation in biotechnology terms. High speed rotation or centrifugation is sufficient to rupture their cell membranes so that all cellular components come out in the buffer. So if it's an animal cell whose genome you're extracting, because your desired protein product is being produced by some animal and you want that protein product at mass scale. For example, you want insulin, and insulin is basically produced by what? Every organism from its pancreas, but everyone produces insulin for their own purposes only, not for humans. Okay, so what you need to do is take a small piece of pancreas of an animal, and that small piece of pancreas of an animal is dissolved in the buffer and then centrifuged so that all the components will come out where? In the buffer. But then, as I told you, if these cells whose genome is being extracted are plant cells or bacteria cells or fungal cells, which means your desired protein product that you want at mass scale is naturally being produced by a plant or a bacteria or a fungus, and you want to produce that protein at a mass scale, then you will require that particular gene from these cells. And to get the gene from these cells, you have to first take out their genome, and that is what we are trying to learn over here. If these cells whose genome is to be extracted are plant, bacteria or fungal cells, then treatment with enzyme. What kind of enzyme? Cell wall digestive enzyme. Cell wall digesting enzyme, that is now compulsory. So we can add, or we have to add, we can say cellulase. If you are extracting the genome from the plant cell, which enzyme do you need to add? Cellulase. or if you're extracting the genome from a bacteria cell walls are made up of peptidoglycans, and who digests peptidoglycans? Lysozyme. Or if you're extracting the genome from a fungal cell, what should you add now? Chitinase, because fungal cell walls are made up of chitin. So if you're extracting the genome of the plant cell, you have to add cellulase enzyme. If you're extracting the genome of bacteria cells, you have to add lysozyme. If you're extracting the genome of, let's say, a fungal cell, you need to add chitinase enzyme. So if these cells whose genome is to be isolated are plant, bacteria or fungal cell, you need to add enzymes, cellulase, lysozyme or chitinase enzymes respectively. That is to digest or dissolve the cell walls. And then we can add a detergent or a solvent, for example SDS, sodium dodecyl sulfate. Sodium dodecyl sulfate actually is a surfactant. Okay, I mean, it's a kind of, let's say, detergent. It's a cell membrane dissolving reagent. So first we digest the cell wall, and then a solvent like SDS is added to rupture the cell membrane also. So that all cellular components come out in the buffer solution now. Because cell wall is digested, cell membrane is digested, everything comes out in the buffer. What do we want from all the cellular components? Only DNA. But DNA is always associated with some proteins like histones, for example. So in the same test tube, we need to add proteases. Proteases will help us to digest all the proteins associated with DNA. And DNA is not the only nucleic acid in the cell. There is RNA also. So I will add RNase as well. RNase is to digest the RNA of the cell. So after taking out all the cellular components in the buffer, I'll add proteases and RNase also to the same test tube. Addition of proteases and RNase is also done in the test tube, so that the only nucleic acid which remains intact is what? DNA. And this DNA will be pure DNA now without any proteins. So over here, if you see, I can just centrifuge, or I can add cell wall digesting enzymes. Then I add cell membrane digestive solutions like SDS, and all the cellular components come out in the buffer solution. Then I add proteases and RNases, so that the only nucleic acid that remains here is DNA. Okay, clear till now? If DNA is the only thing that is remaining over here, how do I basically make sure that I can see that DNA so that I can basically take it out? Let us see. Once protease and RNase treatment has been done, the next step will be addition of chilled. Chilled basically means less than 4 degrees Celsius over here. Absolute. Absolute means more than 95% concentrated. Ethanol. Ethanol is the consumable alcohol. You get it in the alcohol shops also, but the ethanol quantity in alcohol shops, like in let's say whiskey, rum, that is around 42.5% only, maximum alcohol concentration. The rest is water. The ethanol that we are using in biotechnology, however, is absolute ethanol, more than 95% concentrated, and we'll be using chilled ethanol in this case. So addition of chilled absolute ethanol is now done in the test tube. Okay, and once we actually add chilled absolute ethanol in the test tube, this causes immediate precipitation of DNA in the form of yellowish or white threads. In the test tube, where all the components are there, as soon as you add chilled absolute ethanol, you would be able to see a few yellowish thread-like structures. What are these threads? The precipitated DNA. So the most important question over here is the one that is most common in NEET exam. Addition of which reagent is responsible for precipitation of DNA? It's a chilled absolute ethanol. Okay, so that's it, guys. This is how DNA is precipitated out or separated out from all other components of the cell. Once you have the DNA strands formed, and you can see them with the naked eye, okay, the DNA strands that you can see over here, they are visible with naked eye. And what you can do is basically just simply take a glass rod, put that glass rod inside over here, and pick out these things, just like taking something out of let's say a cup with the help of a spoon. So you can do that. You can take a glass rod, and with that glass rod you can basically take out these DNA strands and maybe basically do what? Put them in a separate test tube. Okay, this method is called spooling method. More about this spooling method, more about further processes, we will continue with that in the next video. Okay, so what we have done today is basically obtained the DNA of an organism who is the natural source of our desired protein product. This is obviously just the first step in the story. This DNA needs to be basically broken down into genes, and then that particular gene, which is the gene of our interest, which will be coding for our desired product, that needs to be obtained, and then we have to utilize it further also. So all this in the next video. Till then, just go through this. If there are any questions, feel free to contact me through message, comments on this video, or WhatsApp messages. All of you have my number. So that is all for now. Thank you so much. We'll continue.